Welcome to the video for this week's Mendelian Genetics Lab. In this week's lab, we're going to review some concepts regarding Mendelian crosses. These are concepts we've covered in lecture, and we want to make sure we have them together so that we can go through lab efficiently. We're also going to then go into lab with our laptops this week and do some crosses with Mendelian traits in Drosophila. This is the fruit fly. So to participate in this week's lab, you're going to make sure to complete this online genetics video. And while you're doing that, you can be sure to print out and fill out your completed worksheet and bring this completed worksheet to lab as your ticket to get in. The objectives of this week's labs include first becoming familiar with basic notation for genetics so that we can discuss the inheritance of traits. This will include both standard Mendelian notation, as discussed by Mendel in his investigations of peas, as well as notation that's applied to Drosophila, which is considerably different, and the ability to understand how Drosophila notation revolves around mutants, both recessive mutants as well as dominant mutants. Second, we're going to make sure we can understand the results of simple monohybrid crosses, that is, the inheritance of a single trait. So we'll be able to see how a single trait is passed on from one generation to the next, both again with Mendelian notation as well as being able to do this in notation associated with Drosophila. Third, we are going to expand on that to be able to understand the results of Mendelian dihybrid crosses, that is, when we have the independent inheritance of two different traits at the same time. So it's going to be a little more complicated, but we're going to be able to work through these types of crosses as well this week. So of course our story begins with Gregor Mendel. We've gone over Gregor Mendel, the Austrian monk, who was fond of growing plants and studying their inheritance patterns. Most specifically, he was working with pea plants, and he would grow and record the inheritance patterns within these pea. So he took true breeding varieties, which he called the parental generation. In this case, we have round-seeded variety being mated with a true breeding wrinkled seed variety, and as we recall from lecture, that would produce round-seeded offspring in the first generation, which he called the F1 generation. Because the round-seeded phenotype was the version that showed up in the F1s, he was able to identify a dominant form, the round-seeded form, and a recessive form that would disappear, that would be the wrinkle-seeded form. From this, he was able to generate his notation to indicate dominant alleles and recessive alleles. So in the parental generation, recall that we had dominant homozygote crossed with a recessive homozygote to produce in the F1s a dominant form, dominant phenotype, the round-seeded phenotype, with a heterozygous condition. When he mated the F1s together, in this case two heterozygotes together, what he found was a 3-to-1 ratio of the phenotypes round seeds to wrinkled seeds, for example, in this case. Now we can associate those phenotypes with specific genotypes. In this case, the dominant form is accounted for by the dominant homozygote through a quarter of the offspring, and the heterozygous form through half of the offspring. And the recessive phenotype is accounted for only by the homozygous recessive condition in the genotype in a quarter of the offspring. So the genotype ratio is a 1-to-2-to-1 ratio. Now also recall that for the notation in Drosophila, the notation is a little bit different. Drosophila are fruit flies that were studied by Thomas Hunt Morgan in the early 1900s and also by his students thereafter. And in this case, as he would study these fruit flies in the lab, because they also made a very good model, much like peas were for Mendel, that he could discover the type that was commonly found in nature, which he referred to as the wild type fly, and he could compare them against mutants that were discovered in the lab. In this case, we're looking at a mutant that has a dark or ebony colored body. So he would refer to this as an ebony mutant. And when he would mate the wild type varieties versus the mutant varieties, for example, in a parental generation where you have true breeding forms of each variety, he would find that in the F1 he'd get with the wild type variety in this particular case. That would indicate to him that in this case wild type was dominant to the ebony condition. The ebony mutant was what he referred to as a recessive mutant. And so as a result, his notation reflected not only the mutant but also the mutant's condition with respect to dominance. So the ebony mutant, as a recessive mutant, was named after ebony, E, and given a lowercase notation because it was recessive. The wild type in all cases are indicated by having a plus. In this case, we can say it's a wild type with respect to ebony, E+, or we could just simply say plus plus, recognizing that it's a wild type. So now, this cross was a wild type homozygous parental form versus a ebony homozygous parental form, and they produced the F1, which are heterozygotes as expected. And in this case, we've indicated with the special Drosophila notation plus E. For dominant mutants, such as the curly-winged mutant, the notation works somewhat similarly. In this case, the curly-winged mutant, mated with a wild-type fly, would produce F1s that are all curly-winged, indicating that the curly-winged mutant is a dominant mutant. In this case now, because it's a dominant mutant, the mutation will be named after the curly-winged condition, but it will also be given an uppercase notation to indicate its dominant condition. The wild type will be indicated by having a plus either associated with the mutation notation or simply plus plus to indicate its homozygous wild type condition. So here we have the homozygous dominant curly-winged mutant mated with the homozygous recessive wild type in this case. And that produces F1s that are heterozygous just as expected. We can also have sex-linked loci. And recall that for sex-linked loci, we have to use yet another notation. In this case, we're looking at the white-eyed mutant in Drosophila, and this is a recessive mutant. Here, the white-eyed female is mated with the red-eyed male, and in this mating that produces an asymmetric result according to gender, in which all of the daughters are red-eyed and all of the sons are white-eyed. In order to indicate this through our notation, we have to indicate that this is a sex-linked gene on the X chromosome, and we do that by putting a large X. And then we put a superscript for the actual mutation allele itself, in this case a W. It's a small w because it's a recessive mutant for white eyes. So the homozygous recessive female will be mated with a male who is indicated by having the wild type allele and the Y chromosome. This condition is known as a hemizygous condition, and he has the hemizygote. And for the daughters now, the daughter's notation is that for the heterozygote with a wild type allele and a white-eyed mutant allele. The sons are a hemizygous condition with the white-eyed allele and the Y chromosome. We will also look at the case where we have inheritance of multiple loci, and in Mendelian genetics, this results in independent assortment. So in this example where we have a white cat with a short tail mated with a brown cat with a long tail, we're going to examine these two different traits, tail length and coat color. And when we make this cross, we can use standard Mendelian notation. When we have the parentals cross with each other, homozygous for each of these conditions, we'll produce F1s that in fact are heterozygous for each condition, and that will indicate the dominance of the brown fur and the dominance of the short tail. When the F1s are mated together, we'll notice that we'll get the very common Mendelian ratio that we expect from independent assortment of a 9:3 to 3:1 ratio, and that we can account for these phenotypic ratios with some genotypic indications. Note here that in this case I've indicated that the short-tailed brown-furred condition occurs whenever you have at least one dominant short-tailed allele and one dominant brown-tailed allele. And we've put an underscore in the place for the other allele because it doesn't really matter whether it's a capital S or a lowercase s or a dominant brown allele or a recessive white allele. Because once you have the dominant forms in here, you're going to get short-tailed and brown condition. So this is a new way of generating a notation for the genotype that accounts for all of these phenotypes. And we can simply work our way through the phenotypes, applying the appropriate genotype. We're going to pull out our laptops and we're going to examine an online Drosophila lab where we'll be able to make crosses in a virtual lab environment. So we're going to spend some time becoming familiar with this lab and how to operate it, and then we're going to evaluate some conditions of some basic Mendelian traits that are found in fruit flies. And so this week, as well as next week, you're strongly encouraged to bring your own laptop if you have one, because I'm sure it's going to be in much better condition and you're going to be more familiar with it as we go through some of these online activities that we'll be doing this week and next week. So let's take a look at the online site where we're going to be examining some crosses with Drosophila. When we come to your site, you'll have instructions in your lab handout how to get here. You can simply enter as a guest to get in. Once you're there at the lab bench, you'll notice there's a dissecting microscope, there's an incubator, and there's a computer, and we'll be using all three of these instruments as part of our activities today. So let's examine how this is going to work in the online lab. First thing we'll need to do is go to the computer to order the flies that we want to order. So we click on the computer, and then we can go over and we can order the flies. And in this case, I can order either female flies or male flies. So what I want to do then, say I'm ordering female flies, is I might say, let's order a wild type female fly. So it tells us that it's a wild type here, and it tells us a symbol for the wild type condition. And I simply put that fly in my cart. Now I'm likely going to want to compare my wild type fly against a mutant fly. So for the male, I can go over to this list on the side here and examine the various possible mutants. There's mutants for bristles, body color, antenna, all sorts of things of that nature. So in this case, I'm going to pick a mutant that has to do with wing size, and I'm going to choose this apterous mutant condition, which means they're basically missing their wings. So I select that condition for the male, and I put him in my cart, and then I can go to my shopping cart, and I can check out and buy my wild type females and my apterous males. And here we go, my box has arrived from the company, and I simply click on the box. Notice that I'm following the directions here that are on the screen, and when I click on the box, my virtual assistant will unpack them and put them into the incubator in order for the incubation to begin and the flies to be happy. My virtual assistant has made a mating jar then, and when I pass over the mating jar, I can see that in fact what's in here are wild type females being mated with apterous males. So I click on the jar, and they put them in the incubation chamber, and the flies begin to mate and breed for a couple of weeks. Now fortunately, we don't have to wait a couple of weeks like we used to in the old days when we actually used real flies. Now the flies are ready to examine. So I can open the incubator and I can click on my mating jar. And what we'll do is my virtual assistant will knock the flies out and put them under the scope for us to examine. So here are all the different flies, and we're going to have to sort them out by gender and by whether or not they're a wild type or a mutant. So I simply click Sort Flies. And there goes my virtual assistant sorting out the flies for us. And they put in two piles, and when I roll over the pile, it tells me I have a number of females here who are all wild type, and I have a number of males here who are all wild type as well. In fact, up here on the right hand side, I can see what my results were, and that they were 622 female wild type and 614 male wild type. So about equal amounts of males and females, and all of them were wild types in the F1s. And that's to be expected because that tells me that wild type is dominant to the apterous condition. I can then go over here and make an F1-F1 cross. So I'll click on my females. I'll get a good close look at one of them, and I can say, please use this female in a new mating. She's my F1. Add it to a new mating jar, and I can zoom out. And I can go to my F1 males. I'll click in, take a look at them really closely, and I can use them in a new mating as well. And then I can zoom back out. And then when I'm ready to do my second mating, my F1 crossed with my F1, I'll return to the lab, and there, lo and behold, is a mating jar prepared by my virtual assistant with the female wild types and the male wild types from the F1 generation. So I click on the jar, I put it into the incubator, and again, the breeding and mating ensues and the babies are produced. Such that in a couple weeks later, I can go into the incubator, I can look at the first cross results and take a look at them again, or I can go to my new cross and I can pull those out. And again, the virtual assistant will put them under the scope for us to examine. And now when we look under the scope and I ask these to be sorted, we find there are 4 piles. We find that most of them are in fact wild type, both wild type males and females, and some of them, a smaller proportion, have the mutant condition. So what I'm going to do is I'm going to send this data that we've generated here to the computer so I can analyze it further. I simply say send to the computer, and then often when I send it to the computer, I just want to go to the computer right now, which is the case here. So now I can go and analyze my results. And what it shows me are the results of the different offspring in the F2 generation. So one thing that's nice, and that in this case it's nice, is I don't have any differences between the sex. The wild type numbers are about the same for both males and females, and the mutant numbers are about the same for both males and females. So this is telling me I don't have a sex-linked gene, I have an autosomal gene, and I can click Ignore Sex. And so now I can do a nice comparison then of the amount of wild types to mutant types. And when I look at the proportions, it's not exact because we're only examining about 1,000 individuals here. But what we find is it's almost, or it's very near to 0.75 to 0.25, or a 3-to-1 ratio. And that was what we would expect from an F1 crossed with an F1 in terms of the phenotype ratios that are here. So now I can return to the main menu, or I can add this to a notebook where I keep track of what I've been doing. But we're going to record this in our own laboratory handout in order to compare our results to each other. So if I return to the main menu, I can either order some new flies, I can go back to analyzing the results, or I can return to the main lab where I can again have the various options to examine some of the matings that have already occurred, or again go to the computer and order new results. Now one small issue is that because we've entered as a guest, we're only going to be able to do 3 matings before our incubator becomes full. So when I want to study another trait, when we'll study multiple traits in this lab this week, I'll simply go back out and I'll simply re-enter as a guest again. And when I re-enter as a guest again, I'm ready to go on and order my next set of flies with my new trait that I might want to study. So when you work through the lab handout, we'll work through multiple traits and look at different types of characteristics. Some of them are Mendelian characteristics, some of them might be sex-linked characteristics, and then we'll also look at a dihybrid cross where we're looking at multiple characteristics at the same time. But now you should be familiar with the online virtual lab setup, and when you get to it in lab, you should be able to move through rather quickly. So to participate in this week's lab, you're going to make sure to complete this online genetics video, and while you're doing that, you can be sure to print out and fill out your completed worksheet, and bring this completed worksheet to lab as your ticket to get in.